In bryophytes, the third class is Bryopsida, which includes mosses. Mosses are considered to be the developed Bryophyta group of organisms. These also grow, here also the two generations are there, gametophyte and the sporophyte. The gametophyte is the dominant stage as like in the other Bryophyta group of individuals. So if you see the gametophytic generation, haploid, independent, green, photosynthetic, autotrophic in its nature, which have the organization like this with root-like structures called as a rhizoids, and the colloid structure which have the branched like organization also, and on that we can see the development of leaf-like structures. These leaf-like structures are called as a phylloids. Rhizoids, phylloids, colloid, and at the base, at the tip of one of the branch here we can see the growth of the sporophytic generation, which is represented by the presence of food seta and the capsule. This is a sporophyte generation. So this part is considered to be the gametophyte. Here in this one, we can see the seta and capsule part, whereas the food is completely embedded in the tissue of a gametophytic generation. That's why we can't see from outside, but sporophyte contains food, seta and the capsule. Here if you see the rhizoids, generally rhizoids are brown in color, which are branched. Branch structure is there. Generally they have oblique septa, which will increase the area of absorption. That is, rhizoids are multicellular, darkly colored, which are useful for absorption of nutrients and water which is required by the plants. Then generally colloid will be aerial and it is green in color, which produces branches and leaves. The leaves are called as phylloids, and these are green and photosynthetic in their function. The arrangement of phylloids is also been a specific manner with a specific type of phyllotaxy. Then this gametophyte, when it participates in the reproduction, it produces two branches. Those are called as male shoot or male branch, and female shoot or the female branch. The male branch is called as antheridiophore, whereas the female one is called as archegoniophore. The antheridiophore, which produces antheridia, along with the antheridia they have paraphysis, and this is a branch if you consider. So these are the cells in the male axis. In this one, all these antheridia are surrounded by leaf-like structures. These leaf-like structures are called as pericytial leaves, which are protective in function. And in the middle region we can see the antheridia of different developmental stages. Along with that one we can see some hair-like structures which are green and photosynthetic, and sterile hairs, so which does not participate in the reproductive process, and these hair-like structures are called as paraphysis. And here the antheridia of different sizes or different developmental stages are there. So that's why we count mixed type, where young ones, developing and the matured are present. So they have a stalk here. and surrounded by a jacket. In the middle they contain the antherozoids. So this is the antheridium. The antheridium is generally club shaped, and already we had discussed that reproductive organs or the sex organs are multicellular, stalked and jacketed, and inside which they produce many antherozoids. Generally these antherozoids are motile, having flagella. So in the structure of archegoniophore, here also the archegonia are developing at the tip of the branches. So this is axis of archegoniophore, where here also the archegonia are protected by some sterile structures. These sterile structures are called as pericytial leaves, which are multicellular and protective in function. And in the center they have the sex organs, that is archegonia. So here also the archegonia of different developmental stages can be seen. Along with that one we can see some sterile hairs which are green and photosynthetic, which does not participate directly in the reproductive process, and these are called as a paraphysis. So here the only difference here between the antheridiophore and archegoniophore is, in antheridiophore the antheridia are present, but here in place of antheridia the archegonia are present. Here each archegonium will have flask shaped structure with three parts. The lower part is called as a stalk, and middle swollen part is called as venter, and upper long and elongated region is called as a neck. So likewise three parts are there. Here stalk is multicellular, which help in attachment of the archegonium to the branch, and venter region contains two nuclei, and this is surrounded by two layers of two wall layers. So this is called as a venter wall, and in which we can see a big nucleus on the lower side and a small one on the upper side. The bigger, larger one which is present on lower side is called as axil, and above that one smaller one is called as ventral canal cell, or VCC, and above that one we can see the long and elongated structure. This is called as a neck. So neck is surrounded by some protective jacket layers. Above the venter the long and elongated part is present, that is called as neck. This neck also having the jacket structure with some cells in the center, and the jacket cells are called as a neck cells, and in the center we can see some cells, those are called as a neck canal cells, and above that one the cap cells are also been present. So this is the part structure by which this archegonium is made up of, and one archegonium will produces only one egg cell which is haploid in condition. The antheridium releases the antherozoids when the antheridium matures, and these antherozoids will swim in the water with the help of the flagella, and they are attracted towards the archegonium. Here when the archegonium become matures, the ventral canal cell and the neck canal cell will dissolve and form a chemical substance, that is mucilage will be formed. Because of this formation of the mucilage, the pressure will be exerted on the capsules and all the capsules will open up, and this mucilage like substance will come out in the form of a drop like structure. So this chemical substance will attract the swimming spermatozoids, and many spermatozoids will come and attach to this mucilage, and when it occurs the mucilage gradually dries up. When it dries up, what happens, this entire mucilage will go inside, and in the meantime, along with that mucilage, the spermatozoids will also enter into the venter of the archegonium. Many spermatozoids will enter, but out of all the spermatozoids, only one of the spermatozoa will fuse with the egg cell to form into zygote. That is, the fertilization will occur inside the venter of the archegonium, and the zygote is also been formed in the venter of the archegonium, which is diploid in condition, and this zygote will gradually undergo mitosis to produce an multicellular and diploid structure called as embryo. Embryo formation will also occur in the venter only, and the development of embryo will also occur in venter. In the meantime, the venter wall will also grow and develop along with the embryo, and that protective sheet which is formed from the venter wall is called as calyptra. Initially, the calyptra is acting as a protective covering of the young sporophyte which is developing inside the archegonium. Now, this sporophyte is formed from the embryo which is developing on the female shoot, because the embryo formation will occur in the archegonium which is present on the female shoot. That is the reason why the sporophyte will be there on the female shoot only, and this sporophyte is initially protected by the gametophytic protective sheet that is called as calyptra, and this sporophyte contains three parts. One is foot, seta and capsule. So, if you observe the outline of this structure, so foot is the one which will absorb the nutrition, or particularly water and minerals from gametophytic tissue, and foot is a structure which will conduct the water and minerals which have been absorbed by the foot up to the capsule region, and the capsule will have three parts. Those are apophysis, theca and operculum. Here, if you observe the structure of capsule along with three parts. So, the lower part is called as seta. The seta is extended into this middle portion. So, this have vertically elongated cells. Vertical elongated cells contain region is called as a apophysis. So, this is considered to, this entire part is called apophysis, and the central part is called as a central strand, and the thickness of the wall is there, where it is made up of many celled thickness. It contains epidermis and different types of layers are there. The middle part is called as a theca, which is actually the spore producing region. That means it is considered to be the fertile portion, and here the columella is present along with the spore sacs. These are spore sacs, and central part is called as a columella, and the spore sacs contains the sporogenous tissue. This sporogenous tissue will undergo mitosis to produce spore mother cells. As here this sporogenous cells are diploid in the nature, the spore mother cells are also diploid in nature, and these spore mother cells will undergo further meiosis to produce spores which are haploid in nature, and all the spores which are produced are of similar type. That is why we call it as a homosporous in their nature. Almost all the bryophytes, not only the mosses but all the bryophytes are homosporous in their nature, and here this part is called as a operculum or the lid. This part contains some special structures which are helping the dispersal. So, that is called as a peristomial teeth. So, here in mosses, the elaters are not present, which were there in the liverworts, which are helping in the dispersal of the spores. But here the peristomial teeth are the structures which are helping in the dispersal of the spores, and once the spores are liberated, so these spores will fall on suitable substratum and they germinate and give rise to the gametophytic generation. Gametophytic generation also have two, one is first it will form an juvenile stage. The juvenile stage is called as a protonema, which is leafless and filamentous structure. It contains two types of filaments. One is called as chlorenchymatous filaments and rhizoidal filaments. Chlorenchymatous filaments are aerial and which are green, whereas rhizoidal filaments are non-green and grow into the soil. So the spores, when they germinate, they forms the juvenile stage of the gametophytic generation. So here this protonema which is formed, it will produces buds on it, and these buds, when they germinate by vegetative reproduction process, they will give rise to the gametophore, which is considered to be the actual dominant gametophytic generation, which is having leafy gametophyte stage. That means here the gametophytic generation have two plant bodies, one is protonema and the adult stage, that is called as gametophore. Likewise, once again the gametophyte have been formed. So here once again the life process will be going on. This is a way how this alternation of two generations will be there, and the life cycle is starting with a haploid generation, that is gametophytic generation. That's why we call it as a haplodiplontic life cycle. And the two generations, that is the structure of gametophore and the structure of sporophyte are quite different. That's the reason why this type of alternation is called as a heteromorphic alternation of generations. Some of the examples belonging to this mosses are Funaria, Sphagnum. Funaria is called as cord moss, Sphagnum is called as peat moss, and Polytrichum. This is called as hair cap moss.